Hello everyone. I'm just gonna hold on a minute and wait for everybody else file in. Hope everyone's doing well, finding a way to stay busy but also sane, and hopefully have access to everything that you want and need. Those of us who do are quite lucky. AP Biology Review, Pandemic Edition. In these strange times, we've got a strange exam ahead of us and strange circumstances in terms of making sure we know all this material. We're getting the kind of instruction we're used to in school. So we shall forge on and try to review everything we can. No big deal, just weeks and weeks of class, up to an hour a day, several days a week, condensed down to about 6 hours. No problem. I'm Andrew Dufresne. I'm a senior tutor with Prep Matters. You can also reach me at Mental Markets there. This time I actually thought to put it on my slide. And just a reminder here where we've been and where we're headed. Today is going to be all about the cell. I'm actually very excited about this unit. I think it's really cool to see how everything kind of comes together. I think it leads for a nice segue to the next parts too, to wrap up the units that you need to know for the AP exam. As a reminder, the big ideas that College Board has in mind to connect everything, they're listed here. Evolution, that idea of adaptation and change for version, not my phrase, just in all the randomness, whatever works best. That carries on. Energetics, the idea of energy and how things actually get to that next week a lot more. We've been talking a little bit about it in terms of just likely reactions will occur, or what transmission. So this idea that we've kind of, the cell or the molecules or whichever level you want to look at have kind of figured something out, and they don't want to reinvent the wheel each generation. They want to persevere and have some form of longevity. Then we need to be able to encode that information and then transmit it. That was the major topic the past couple of weeks. Nucleic acids, genetics, inheritance, heredity. And then systems interactions. This is going to be a bigger one today. We have been building up from a zoomed in microscope at the atomic and molecular level, gradually drawing that back. We're now at that level where we're starting to see a community of all of those different molecules and proteins and macromolecules that we've talked about coming together and seemingly like they've all got their job and they're kind of working together. But then there's also interaction with outside that unit. That unit is the cell, and then there's the environment outside. And we've mentioned this before, inputs and outputs. So they've got their big ideas, and then also this idea, this topic of skills of a scientist, or what they consider your laboratory skills, or the learning objectives in terms of looking at data, essentially always come back to make some observations. We see patterns. That's what we're looking for too in this material. That's where how I bring in my themes of structure and function. It's going to be huge today. Directionality and endedness. Smaller elementary particles come together to form larger, more significant ones. We are just basically seeing patterns and then going forward with those. And pragmatically and practically, when we talk about the verbs and task verbs and action verbs that College Board likes to use in their free response questions, so that we know what is it that they have in mind and what is it that I want to do for that answer. And then the hypothesis is essentially the prediction of what's going to happen based on that pattern that we're seeing. And that's huge to basically be able to go from that data that you're given to then say, oh, okay, yeah, I understand enough about this content and this material to know that this would be the next process or action to happen. And then putting together a way test that, describe it, analyze it, graph it, all of those things, and then what conclusions we can make. So we're constantly in that mindset, with that scientific approach. We're thinking about their big ideas, and I'm trying to bring that PMTV difference. The real reason that I know you guys tune in to do some review with us, to basically mention here's a look behind the curtain, here are the different patterns. If we think of this, then we realize that we're actually learning a lot of the same thing over and over again, but it's just taking a slightly different form. And that's fantastic. That just makes us feel more comfortable, more familiar, be able to process it better, and dare I say, even appreciate it and have some fun. All right, so I've mentioned some of these things before, but as a recap, in the event you haven't settled in for a comfy few hours of classes 1 through 3, smaller things come together to make bigger, more complex things. This idea of the molecules and the macromolecules, and proteins. If this weren't the case, we wouldn't be able to talk about what we're going to talk about today. Structure and function, I already mentioned how that's going to be entwined. It is. Everything basically, the function is born out of whatever their structure is. And that structure was something that kind of just happened through random but energetically favorable interactions. And then here we are. And we're going to talk about how we've even co-opted, how cells have co-opted the structure and function of other things to do their bidding. We also then talked about how when this is figured out, let's write it down, let's hand it off, let's find a way to divide cells and to make gametes, sperm and egg, for sexual reproduction, or spores for asexual reproduction. But we need to make sure that this information, we're not waiting for everything to happen again, because it kind of took a long time for all this to basically come together. Last week was a little bit more about alleles and inheritance. I mentioned towards the end, the reason I wanted to mention this again is because I mentioned at the end of that class, in the event you haven't seen it, about the role the environment can play. And the environment is literally all around the cell. It's engulfed in, and there's constant interaction. And so just as what we have for genes and what we express for a genotype becomes our phenotype, that then depends upon the environment. And then inputs and outputs. This is going to be the big ending towards today's talk about transport in and out of the cell. And there's the mention once again of the genetics environment, and then how that exchange, that transport in and out of the cell across the membrane, how regulated that really has to be, and what's regulated. What do we need to know based on the material we're expected to know for the test? Okay, so we are going to start with an overview of the cells. Not going to be a lot of different slides. We're not actually going to be doing the same vocabulary slides that we've done in the past. There's a lot. And as I've mentioned before, I'm expecting you to do some review on your own, because we just simply can't condense everything down. If you do have questions, if you're doing some review on your own, reach out, classes@prepmatters.com, social media, hashtag PMTV. I'm at Mental Markets. I live for questions, and that's not actually an overstatement. It's really the escape that I have from this pandemic. So please send me your questions. If you got questions during today's seminar, today's class, we've got this Q&A function I want to remind you about. Go ahead and throw your questions in there. There's a raise hand function. Do that so I know to look over there. Every now and then I'll check, make sure that everything is clicking, and try to have some questions for you guys too. Yes, we're going to do a review of all the different organelles or smaller components of the cell. And you're just going to want to have an idea of their identity and how their structure and function interact. We're going to talk about the difference between animal and plant cells. AP biology loves that. I mean, it's also interesting in its own right. And then, like I said, we're going to go into transit across the membrane, transport of things in and out. But then also, here we go back to our favorite friend, water. The entire environment of the cell it's aqueous. It needs that for fluidity. It needs that for motion. It needs that so that friction and all that kind of aspect of physics doesn't play a role in getting things to and from. So we're going to revisit water and all the things that it's doing in terms of the structure of the cell and transport of things in and out of the cell. Okay, so why a cell? I would love to be able to have the time to tell you about Anton von Leeuwenhoek, this fantastic Dutch scientist that is the reason that we discovered the cell. He was looking at some cork. He called them animalcules. So an animal molecule. I think that's pretty cute. I feel like that would totally get some hashtag recognition in this day and age. But what cells allow for, what we mean by a cell, is an enclosed structure. All the different parts that we've been talking about. We talked about them a little bit during meiosis, and I mentioned mitosis, but really we're going to be talking about all those different components today. And what the cell provides for is an ability to compartmentalize, to specialize, to really only have to do that one thing, and someone else will do their part and build up. So it doesn't matter the size of the organism, they all come down to similar cells. They differ in size, of course, but there are reasons for that, and we're going to talk about that in a second. So these small things come together with their individual roles. We're not going to get into specialization, but the way this goes, cell, tissue, organ system, organism, that's the usual hierarchy from smaller things to bigger things. We're just going to keep it to prokaryotes, eukaryotes, without a nucleus, with a nucleus, bacteria, not, and animal and plant. Now, I mentioned a second ago about the limit that a cell can reach in size. We're like everything is fair game in the entropy of the planet, the universe, and the environment, but the easiest stuff, the stuff that gets the most done with the least amount of effort, that's going to win out. So we have some limitations to what the size of a cell can be. So let's imagine for a second here that we just have a cubic cell. It's just easier than the weird oblate, spheroid, kind of whatever it is, shape, and also it kind of shifts and changes. But if we were to consider a cell to be cubic, and we could compare different sizes just on relative side lengths. So what do I mean by all that? You've got 1 centimeter on each edge for the small cube, then 2 on the next, and then 4 on the next sides. Now, there's the inside of the cell, and there's the outside of the cell. The inside is three-dimensional and it has a volume, but it can only interact with the outside through the membrane on its edge, which means the surface area of that membrane is going to dictate the efficiency at which things can be exchanged. And there's no way we can live without that exchange. We absolutely have to take food and energy sources in. We got to get rid of waste. There's communication. Do I really need to impress upon you the importance of socialization between cells, considering it's a pandemic and we can't even have human interaction? So we've got the area of the small one would just be 1 squared for one face, and there are 6 faces. So again, we're thinking of the cube, trying to keep this simple. So that would essentially be 6 square centimeters of surface area on that small cube. The volume would just be 1 cubed, which is 1. So your surface area to volume ratio, your ratio of the options you have to interact with your environment with all the stuff that's inside the cell and needs to come and go, is 6 to 1. You'll notice that as you go to a bigger cell size and then another bigger cell size, that surface area to volume ratio decreases. Just by doubling the size once, it goes from 6 to 1 to 3 to 1. It cuts it in half. And then doubling again, same thing again. So it becomes 3 to 2 or 1.5. So we really do have limitations as to what we can do, because we won't be able to, we'll have too much stuff inside and not be able to get stuff in or out, or we could be too small and basically be so much surface area. We need a particular minimum volume to be able to hold all our stuff. And you're not fitting, 2 or 3 bedroom amount of stuff in a studio. So, okay. So this surface area to volume ratio is something that comes up in free response questions and is a general concept because it's the logistics to why a cell and why that size and all that. There are limitations. It's just one, but it's a popular one for the test. Okay, so there's inside and there's outside. Outside, the entire environment, aqueous environment, all the goodies, food, all the stuff you want to get rid of, waste. But what about the inside? Because that is obviously going to be where the membrane formed around, and we were trying to compartmentalize, and we felt that some special stuff was happening in there. So what exactly is going on? Well, let's start with the prokaryote. There is no nucleus. This is your bacteria and your archaea, the Monera domain. We think that these were basically first. They're simpler. It kind of makes sense. We're building up from smaller things into bigger things. Monera, or bacteria rather, bacteria are an example of prokaryotes, and then the archaea, which were divided out and kind of made to be a different thing, those are usually more the extremophiles. So the extreme environments of the planet is where we find those. Ridiculous pH, acidic or basic, pressure, heat, just general chaos. Makes sense that you'd want to be a simplified form, but still have enough going on to be able to pass something on. Okay, so what do we have here? The cytoplasm is really that majority of space inside. It is not empty space. It is fluid filled. And there's a slurry of all kinds of other things. You got proteins swimming around, molecules and all that. So that's really just the interior. There is no nucleus, like I said, but there is a nucleoid or a general area where all of the nuclear information, or the genetic information, is stored. We remember, hopefully, that in prokaryotes and bacteria, they don't have multiple chromosomes like us. They have the one singular circular chromosome, the plasmid. Why they didn't draw that, I don't know. This is, as I've mentioned before, the free stuff that I can find that gets us to where we're going and is so much better than my art. Okay, so apart from that, is there a lot going on in prokaryotes? We wanted to stay simple. We've got the genetic material. What else do we need? We need proteins, so we need ribosomes. Ribosomes are where we're making our proteins. We talked about those class 2 about nucleic acids and translation, where the RNA comes in and then proteins come out. And then other than that, I think we got everything we basically need. We just need to protect this and guard it from the environment. That's the plasma membrane. We're definitely going to be talking about that more. A cell wall is not uncommon in prokaryotes. It's usually some kind of mesh of protein and sugar. Sugar is another, we talked about that molecule in the very beginning. There's a reason, because it's important. And that's often presented on the surface as well to signal to the environment. And then this capsule, this is another specialization that bacteria made, prokaryotes, but it's just extra protection. So it's another chemical layer. You're not going to have to know too much about the structures of each of these things, just basically, okay, the general structure is on the outside, and their function is to protect. There's this one other thing that I really like that's in this drawing, and I want to draw attention to, and that's the mesosome. That folded area of membrane right here. This gets back to the surface area and volume ratio we talked about. By having lots of folds like this, why am I spending so much time on this? Because it's going to be important, it's going to keep coming up. By having so many folds like this, in that same volume, that same space, we have a lot more surface area of membrane. So you get a lot more exchange. This is key, even in these guys, to essentially have the production of energy. This is going to look a lot like a lot of structures that you've heard about, that you've seen before, and that are part of us. Okay, so simple cell structure, it's got the basics, it's good enough, prokaryote. Now enter the animal cell. Lots has happened since the time of the prokaryote. The prokaryote is obviously still around, but a lot has happened since it was just that. Because now the animal cell not only is bigger, but it has all of these different organelles inside, all these specialized compartments, these structures that have one job, and with the dance of everybody, the balance of every one of them, they manage to achieve much more and be much more advanced and detailed. Now, I'm just going to jump right to the chase because I mentioned the folds of the lysosome and the prokaryote. Perhaps does this look familiar? Perhaps does a multi-stacked membranous organelle like this look familiar? Or the endoplasmic reticulum? These are all membranous or lots of folds, high surface area to volume ratio components here, and they all have specific jobs. So in fact, there is the theory, the endosymbiotic theory, that you likely covered in your class when you went over this unit. Basically, mitochondria, these blue guys here, notice that there is more than one. We're not typically talking about a mitochondrion. There are going to be even more of these as the producers of energy for the cell in things that need a lot of energy, like muscle cells. But the mitochondria are likely bacteria that enslaved by the animal cell. So this outer membrane and then the intersomic vicinity, and then made that, okay, you're just going to do your job that you were doing, you're going to churn out ATP, which is our energy currency. We're going to talk more about that next week, a little bit later at the end of the day here today. But that's basically what we think those are. And that's the case for a lot of these organs. So the ribosome is notice the size difference here. When we go back to here, they're, granted, this is drawn to scale, not necessarily, but there's a lot less going on here, so the ribosome is a major player. And then here, oh, it's just this little grain over here. So those are made up of protein and RNA. Those are not something that we took from another prokaryote and had a specialized job. That was something that came together structurally and chemically and did its job, as it did in prokaryotes. But lysosomes, peroxisomes, potentially Golgi and the endoplasmic reticulum, these, a lot of the organelles may have come from prokaryotic origins that were then taken in, and talk about how that happens at the very end, and put to work inside the cell. So what do some of them do? We've got now a nucleus, the very center of the nucleolus, or where all the genetic material is. It's also where we're making that ribosomal RNA, that very important RNA that's going to allow us to make all of our other proteins, because it's just the way its chemical structure works and the way it interacts with proteins. The general cytoplasm inside the nucleus, nucleoplasm, and the nucleus itself has a membrane in order to further specialize it. Remember, we talked about how mRNA had to be, you need a 5' cap and a poly A tail. It needs to be modified in order to signal that it can then export from the nucleus. Regulation, so that things are very precise. And then just like our outer plasma membrane, we have pores in the nuclear membrane. Where should we go next? We didn't talk much about the flagellum and the prokaryote, but I like to bring it up as something that's also entirely possible in animal cells. So not only on very tiny, microscopic, unicellular animal cells, but cilia, like the cilia pluricilium, they're basically these little hairs that can come off, or structures that can come off of the cell, and they're usually numerous as opposed to the larger flagellum. But both the flagellum that we saw on prokaryote on the previous slide and the cilia here, they still exist in our cells. Sperm cells are a great example of having a flagella to aid in its motion through an aqueous environment. And cilia, well, they're all over our intestines, and even our alveoli and our lungs. in order to basically do what they do, wag and move around that aquatic environment in the vicinity. But what that does is in our intestines increases surface area, and in our respiratory system keep it clear of dust and particulates. So again, we're just talking these smaller things coming together and doing their singular part, their singular role, and achieving much more. The sum is greater than the parts. So the lysosome, what's going on in there? We're breaking things down inside. It's a bunch of catalytic enzymes and usually an acidic pH, but that's where things go to just dissolve and be broken down. And there may have been some extreme prokaryotes that have that role, and then we're taken in by the animal cell. Centrioles, we mentioned these. They're up here, the cylindrical things here. We mentioned these during meiosis. Their main job is spindle formation, pull the chromosomes to either side, make sure that when we're dividing, we're doing it well. Very important job. The centrosome, the general region where you will find those. The peroxisome is like a lysosome, but focused on hydrogen peroxide and producing breakdowns of that, products from breaking down hydrogen peroxide. The rough ER, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, two distinct functions here. Now the rough, that's where basically ribosomes attach to that, and through this channel in the cell, it's all internal, that creates, it's where the proteins are made. That's protein synthesis right there. And the smooth ER, very fittingly, is all the non-protein synthesis, so lipid synthesis, fats, and macromolecules that are not protein-based. Those are actually generated on the smooth ER. And then now we've got this Golgi, which is probably one of the weirdest, but also very important. If something's not important, it's not going to stick around. There's plenty of other organelles. These are the ones that you usually need to know. But this is making vesicles. This is taking those proteins we just made on the rough ER and helping them fold properly, speeding that up with enzymatic activity. And it basically, it buds. It goes through the bottom to the top stack. And this can sometimes end up packaging something that then gets exported. So it's the suitcase manufacturer, along with folding proteins properly. So all of these are organelles that you're going to want to be familiar with for the content that can be fair game on the free response questions. But again, I also want you to see how they all kind of make sense in terms of what their precursors did and then what they do. Some of these are new, of course, the endoplasmic reticulum, the Golgi, the lysosomes, peroxisomes, but their name can also help you remember. Lys, break down, so that's the lysosome. Peroxisome, peroxide, hydrogen peroxide. I got nothing for Golgi. But all right, so that's an animal cell. What about a plant cell? So there's a number of differences because their functions are very different. So their structure is going to be different. The plant cell, most notably, I'm going to stick to the major differences. In addition to the plasma membrane, they have this cell wall. So the cell wall is much like the cell wall of the prokaryotes, but it has more rigidity than that plasma membrane. So there's going to be added protection, but it's also it's going to be very functional in terms of how and why that rigidity plays a role. So what is that typically made out of? Cellulose, which is a sugar. Cell walls also exist in some non-plant cells that are eukaryotes, fungi, and then so those have a cell wall, and that's usually made of chitin. Fungi are what we call heterotrophs. They get energy and food from outside, from another other. Autotrophs are plant cells. So they make their own self, so they make their own food energy source. That cell wall in fungi, and I'm trying to bring these things up. You can pause, come back, whatever, but I'm also trying to mention these topics so that you know to pay attention to them in your review, those cell walls are actually made of chitin, which is a different polysaccharide, but another one nonetheless, like cellulose. And chitin, on top of that, it's not just fungi. That's actually also a major component in the exoskeleton in arthropods, in crustaceans and insects. So again, the similarity in the way in which these structures are just like, ooh, I like that, I'm going to use that. Ooh, I like that, I'm going to use that. Even though we've got different environments, there's enough similarities where if I just modify this a little bit, boom, it suits my purpose. Okay, so I mentioned plant cells making their own energy, not just the idea of mitochondria, which really comes from making energy from breaking down sugar, next week's topic, but chloroplasts. This green guy up here, that's where our chlorophyll would be. Again, we're going to talk about more about this next week for energetics, but that's going to be an additional powerhouse inside the plant cell. And then we also notice, looks as though it may even be bigger than the nucleus, and at times can be because it's the major organelle in plant cells, the vacuole or central vacuole. It's usually one large primary one. You'll have some smaller ones, but they usually have this bigger one here. It's like a vesicle, but it's empty or looking to have stuff that's actually never empty because it always has fluid in it or food or waste or anything else. It's that storage location. So, you know, plants and plant cells have a different interaction with their environment, a little more staying in place and less interaction with other. So a lot more storage, but also it's the big player in water being taken up. And if you recall a limp or flaccid plant leaf versus one that is stiffer and upright, drooping versus defying gravity, that's usually because the water pressure inside. When it's defying gravity and it's firm and upright, then it's full of water, and that central vacuole would be very large and full of water. So those are some of the major differences between animal cells and plant cells. Again, check out these images, make sure that you can describe the different organelles. You're going to be fine in that report. Okay. Prokaryote, animal cell, plant cell, talk about the various components inside that we're trying to protect and organize further by keeping them in close proximity. What is our barrier between that very comfy, cozy inside space and the harsh, real environment on the outside space that we still need a lot from? And that's the phospholipid bilayer or plasma membrane. And so the phospholipid molecule that we mentioned in the very beginning, class 1, we talked about the molecules and building blocks for life, comes back. And as we know, if you haven't watched class 1, here we go, the recap. Polar head, and then non-polar tail. Hydrophilic head, loving water, hydrophobic tail, hating water. So those are going to naturally want to show the hydrophilic head towards the aqueous environments on the inside and outside. We have extracellular fluid here up top, so the outside of the cell, cytoplasm, the inside of the cell down here. We want those hydrophilic heads to be facing towards the environment they prefer, and then the hydrophobic areas, those can interact with each other and be spared that are butting against water, which they do not like. And that is extremely important. That allows so much regulation of in and out, and is really why something can increase in complexity. Energy in, waste out, keeping a very particular concentration of various ions, homeostasis. This is a word you're going to have heard a lot in class. You're going to want to understand that concept for the test as well. The maintenance of the ideal living conditions. So the give and take as things are used and broken down in order to keep things just right. Okay, so what's going on in this membrane? We have it's riddled with proteins. This is what this combination of proteins these integral proteins, peripheral proteins are on the periphery. Integral, they're integrated, they're all the way throughout. Surface, they're on one side. Glycoproteins, they have glyco or sugar attached to them. Globular proteins are basically just the general way in which biologists draw complex chemical structures. Channels, so transport proteins, we're going to talk about these more. All of these are combining with the plasma membrane to make a fluid mosaic model. They give some space between the components of the membrane, and they also kind of keep things together. So if we notice in terms of some of their structure and how they manage, like why do they have different areas? Well, because of their hydrophilic and hydrophobic amino acids that make up their structure. So you can see here, there could be an integral protein that had alpha helices in the middle, which would be comfortable with that hydrophobic region, and then the hydrophilic regions on the inside and outside. So there's one other thing that's pretty big here, and this is cholesterol. Before I go on, I want to address a couple of questions I've got. Do animal cells have vacuoles? Basically, yes, because they are vesicles, and those vesicles can hold all sorts of stuff. It's nowhere near the size of the plant central vacuole or anything like that. But it's not something that's unique to just plant cells. It's such a bigger role in plants. That's why I drew more attention to it. And also another question: integral proteins, do they aid in transport of materials? Yes, they're extremely important. One such integral protein would be this channel one, and there are others that are transporters and exchanging from one thing to another. We're going to talk about those later. So back to, thank you very much for those questions. I really love it. Cholesterol. I mentioned this as a macromolecule, or a molecule rather, in the very beginning. And obviously it seems to be a little bit different than these phospholipids, but seems to align pretty well with a hydrophobic region and a hydrophilic region. Well, the structure of cholesterol aids in the function of the membrane, so naturally it's going to find a home there. So those hydrophobic tails, remember we talked about saturated and unsaturated hydrocarbons, which are those nonpolar regions, these yellow tails. If they're unsaturated and they've got a double or a triple bond in there, then they're going to have a kink, because they're going to have a bend in them, because they don't have the same, they're shorter and they're just more rigid, and they don't have the same bendability. So if that is the case, if that were to happen too much, in order to get some space between the phospholipids, then it would be too fluid. It would basically fall apart. But if we don't have anything separating them, then they're really compact. And that's going to make it harder for things to come in and out. Even with the proteins, the integral proteins, when those are inserted into the membrane, I mean, it's going to be easier if it's not so packed in the first place. So how do we, what randomly happened, it happened to work and stuck around? Cholesterol intercalated into the membrane, it entered into the membrane. And when it did that, it basically provided just enough of space between the phospholipids, and it worked out in terms of hydrophilicity and hydrophobicity. The other thing that it allowed for was a greater range of temperatures that would work out okay for the cell. So at colder temperatures, cholesterol prevents the phospholipids from compacting too much and getting too rigid. So it keeps some fluidity at a colder temperature. And at a warmer temperature, it reduces that fluidity, and it keeps them associated with each other, and keeps them from kind of basically degrading as quickly, just because of the microenvironment that it creates. So that worked. It broadened the range at which these cells could operate and maintain their composure. Okay, So more about those proteins. This is the big thing that we've been talking about. You had some great questions leading into it. What's the cost of entry? How do I get inside? How do I get outside? It all depends. The two big things that you're going to want to know. Remember, I don't have the vocab slides anymore because there's all this great. I thought it was a great throwback to use Encyclopedia Britannica, something that as a hard copy guy I grew up with. But this is before the internet, of course. All of the terms on here, I tried to get you guys images that would condense it down to just know these terms. This is enough detail. If there's anything else that needs to be added, I'll add it. But if I put too much out there, I don't want to overwhelm you. You're already showing up to this class. You're doing your work. You're doing your review. I want you to know what you can spend your time on for the best impact. So the big things you got to know here are passive transport and active transport. Passive transport is the simple diffusion, facilitated diffusion on the left, and then active transport is obviously this one. We're going to talk about it a little bit, just mention the difference between primary and active. We'll come back to active transport. Well, let's first start on this side here. So already we can probably tell from this drawing that we're talking small molecules for simple diffusion, and then perhaps some larger things, or things that don't get along with this middle ground here. And that's really the big gatekeeper. We're going to talk about all of this later as well. So for passive transport, the cost of entry, free of charge. So that's both a terrible dad joke, because the most often the things that can simply diffuse through are non-polar, so they don't have a net charge. They're okay going through the hydrophobic tails in the plasma membrane. They're already in the aqueous environment. They're diffuse enough outside the cell to be fine there, and they could be fine inside the cell. It's the middle ground of hydrophobic tails that's harder to get through for a lot of things. But non-polar, hydrophobic, small molecules, those can diffuse through. And for the most part, some steroid hormones, carbon dioxide, and oxygen. Polar stuff, small molecules, but polar and all that need a little help, something like water, they would need a channel protein, an integral protein that forms essentially a channel, just a passage through. This is called an aquaporin. If you want to remember that, you could go with one of two things. A great little story I've got about how the scientist Peter Augrey discovered the aquaporin while doing just the amazing, luxurious research on diarrhea. And basically, the transit of water was so different in the intestinal tract. So thank you for that, Dr. Augrey. But you could also just go with the name aqua, water, porin, pore. So it's the water pore. It's how water comes and goes in and out of the cell. So this is all passive transport. It doesn't require an import of energy. Why is that? I keep saying diffusion. Diffusion is that idea of going from a high concentration to a low concentration. Think of it like the more you're around things that are just like you, the more likely you are to get bumped and pushed out of the way into an area where there are less of you, or there are less of what's anything. So you're going to go from an area where there's a lot of something naturally to an area where there's not a lot of anything. Also, you're going to have a lot of interactions and a lot of reactivity when there's a lot of something around, and then stability, lower energy. What we prefer again and again and again in an area where there's not a lot of stuff. So diffusion is from high concentration to low concentration. When water does this, we call it osmosis. And it naturally wants to go from a higher concentration of itself to a lower concentration of itself. It's kind of weird to think about water having a concentration. It doesn't really. It's the amount that it makes up in that general region. So if there's a lot of solute, there's a lot of something dissolved in the water, then there's less water in a given space as a portion of the whole of everything there than if there were a dilute solution where there's not a lot of solute, then there would be more water in a given space there. And so that means that water is going to flow from more dilute solutions to more concentrated solutions. until it essentially reaches that level. Not necessarily that they're equal. I mean, that's the potential, but there can be other regulation that we want to maintain a particular concentration difference inside and outside. Hint, yes, we absolutely are doing that, depending on the ion, depending on the actions that we're trying to take. So water going from dilute to more concentrated solutions, osmosis or homeostasis. If you have any questions, please just speak up and let me know. I know it's a lot of content, but I'm having a lot of fun sharing with you. Okay. I spent a lot of time just talking about water, and I tried to keep it nice and comfy with images. Now we got to talk about a little bit of calculation that you're familiar with. Again, a lot of times I throw terms at you just so that when you see them in the free response questions, you know what's going on. Water potential. Psi, I think. At least it looks like a psi. Like Raphael, Teenage Mutant Ninja Turtles. I don't know, like that's like super dating myself, right? Water potential is essentially calculations that we do to determine the likelihood that water would move. So this right here is the likelihood that water would move from a location. That's really important to think about. And you're gonna come across water potential in your review. And there's gonna be a lot of different ways you can do it. And I tried to really boil this down to what my students tell me makes sense to them, but also is enough of detail for you to be able to hold your own in written conversation about a topic. And there's all kinds of different orientations of terms that we can use to describe this the flow of water. So that water potential is the likelihood of location. So the higher the potential is, the more likely it's going to move from there. Not to there, but from there. That is made up of two things. Solute potential, essentially the solute concentration. Now that's interesting, because I said the higher the water potential, the more likely it's going to move from to there. And if you figure that out in just the 2 seconds that I mentioned solute potential, you've already got this topic locked up. But if you didn't, we're going to come back to that. We're gonna follow up on this. There's a calculation. It's like a nested calculation that you have to do here for solute. Super basic, and it's got some familiar players. Solute potential. So literally, what pressure is the water feeling? If it's being pushed on from one location to another, then yeah, the higher that is, the higher it's going to move, the more likely it's going to move from that location. Okay, so solute potential. Try to speed up a little bit because I realize looking at time here, I'm going to find that perhaps I should. But ionization constant, essentially the number of particles in solution. If you do recall in chemistry seeing a little I and it being the number of ions or particles in solution, we call it the Van Hoff factor. In biology, we don't often reference that, but it's essentially the same thing. Big C, capital C there, molar concentration. So the higher that is, the more solute there is in the solution. R, our old friend, the pressure constant from the ideal gas law, PV equals nRT. So you can work out your units here. We're talking potentials. It's going to be the 0.0831 constant. You're not even going to need to know that. You're actually going to get an equation sheet, if it's relevant, that you would typically get. As far as I know, AP indicated that that would be provided and expected when you're doing your free response questions. And then T, temperature. So the higher that is, well, honestly, the more solute can also dissolve in solution. But if the higher this is, the more likely water is going to flow to that region. How the heck is it adding to water potential, which is it moving away from that area? And that would just make it negative. So we're still addressing all of the aspects that are playing a role in the likelihood of water flowing. But the bigger this negative is, the more solute there is, the less likely water is going to. Okay, now let's put this together and kind of see what we mean in terms of an actual situation. So if we have a U apparatus like this, we fill it with water, and we have what we call a semi-permeable membrane right here. You may have heard about these with something called dialysis. This idea of, oh, it's only going to let through this molecule, or it's only going to let through water, and it's not going to let through the solute. That filtering membrane is a pretty common component to these experiments, but it's also the major player in dialysis, that motion of selective solutes crossing the membrane. So if we were to put one there, and then all of a sudden, if we were to put solute on the right-hand side, whatever it is, we're not putting any on the left, we're putting some on the right. Well, now water is going to naturally want to flow in this direction. Because here, we all of a sudden, osmosis, there's more water on this side, less water as far as the parts on this side with the solute. So water flows in that direction to bring them to as best we can for concentrations. But then also we can think of it in terms of that solute potential, which you see here. So the higher that concentration, the bigger the negative, the more likely water will flow to there, the less likely water will flow away from there. Lower water potential on this side, higher water potential on this side. And if all of a sudden we were to also apply, forget the solute, but apply pressure on one side, well then we increase the water potential on this side again by basically pushing on the water and forcing it through. If you were to do negative pressure, well, that's like pressure on the other side. So the water would flow towards the negative pressure. This is actually the pressure version of solute potential, where here there's a lower solute potential, but solute potential being negative, that means that that's better for water potential. It's more likely to flow from there. So it's the negative pressure is the water is more likely to flow there because there's less pressure. We can also still think of this as going from a high pressure to a low pressure, or a high concentration to a low concentration. High concentration of an effort, like an actual force in that area, versus the area where that force is not being felt. That's going to be the natural downhill stream. I cover this a lot because it's just so important to some of the more detailed questions, and also a big component to the structure of the cell. All right, so then we also have this idea of what are we talking for solutes and what happens when water flows in each direction. Hypotonic, isotonic, or hypertonic solutions. Hypo, less. Iso, same. Hyper, more. That's how I want you to remember these. So if there's a hypotonic solution, then the solution has less solute in it than the cell. If that is the case, water is going to want to flow into the cell. In an animal cell, the cell can burst. We actually use this method in order to collect things out of the cell and make them burst in the lab. But that would be the direction of water in a hypotonic solution. Tonic, or in this case, tonicity, it's really about the water potential that we're talking about, solute concentration. So that's going to kill that. But in a plant cell, when we have the cell wall that's much more rigid, the central vacuole fills up with water, but we're fine. We have a rigid, more upright leaf. It's called turgor pressure. The cell is turgid, and it's experiencing a turgor pressure from the water basically pushing against the plant cell wall, the water pushing against the plant cell wall. In an isotonic cell, you have the exchange of water in both directions. So that's normal, homeostatic, less rigid, essentially. In a hypertonic solution, where there's a lot more solute outside the cell, water is going to want to flow out of the cell and into that solution to bring it down because of the solute potential. But that's going to cause the cell to shrink. And that's not good because now our surface area and volume are off. So that's not good either. And this is why this whole process is so important. It has to do with transport. It has to do with water. It has to do with regulation through the structure of the chemicals at the barrier, at the boundary. And it has to do with that math we talked about at the very beginning, the surface area and volume. In a plant cell, we actually call those plasmolyzed. The chances you'll see that word in the test are probably slim, but that's what this means, a shriveled plant cell. The plasma membrane comes away from the cell wall, and you get a general constriction of that cell. That's a very wilted and fibrous cell, that plant leaf that would not look good. Okay, now back to this active transport. We're almost there. We're going to be going a little over an hour today just because I want to give you a practice question at the end. But here we are back to our active transport side. Primary active transport is essentially just the transport of a particular molecule. There is only one other kind of active transport. It is called secondary transport, which is basically exactly what you'd expect. Someone hits a ride. So primary transport for one molecule, secondary transport, active transport, would be if another molecule went into the cell on the back of the energy of that other molecule. So what exactly is going on here? The big difference, ATP is getting converted to ADP and phosphate. We're removing a phosphate from adenosine triphosphate, like our nucleic acid before. This is the ATP that would be incorporated into DNA as well. It's got a lot of energy in that last phosphodiester linkage. I promise the things always come back around. And when it breaks it, it provides energy for something else to happen. We're going to talk more about that next week. I do want to talk about an example of it, though, because it's the big thing about transport, homeostasis, and concentrations on either side of the cell. The sodium-potassium pump is really the character that you may be expected to know a little about. Essentially, it requires energy to exchange sodium for potassium. It's all conformational change based on the chemical structure of the protein in the membrane. So sodium comes in, and upon 3 ions bonding, an ATP also bonds to the protein. There's catalytic activity. A phosphate is broken off. That is enough to allow for a conformational change in the protein itself, open on the other side, and then the sodium are going to naturally want to go to, well, they're not actually going to naturally want to go to the other side, but they are going to go to the other side. Now, what do I mean by that? Well, the whole point of this, the use of energy, is because we're going against the flow. Everything before was that idea of high to low, downhill, instability to stability, low energy state, natural inclinations. Active transport works against concentration gradients. Whenever you're going from a low concentration to a high concentration, that's going to take energy because you're going against the grain. So the sodium ions, the conformational change in the protein, that is what actually propels them out into the outside of the cell. That same conformation is now well suited for some potassium ions. This is not secondary active transport because this is the purpose of this transporter, but the potassium comes in, bonds to the protein, the phosphate gets kicked off, that leads to a conformational change, and then of course it releases the potassium inside the cell, which you can bet must be a higher concentration because we must be going from low concentration to high concentration to need to do this. Do you need to know those concentrations? No. You'll probably come across them in your review, but it's not something I felt the need to cover here. Okay, we're going to wrap up now and mention the rest of the ways that things can come and go in and out of the cell. Let's focus on coming into the cell. Endocytosis. Endo in, what you're putting in. Endothermic reactions, you put heat in. Cytosis, cyto, cell, and then just the motions that the cell's going through. How's this working? There's a few different kinds. If it is solid and larger in size, we call that phagocytosis. The plasma membrane, it says, forget the whole idea of proteins helping me out here. This thing is huge. I'm going to literally just kind of engulf it. This is actually also how unicellular organisms would consume stuff, like amoebas would consume stuff to bring them into the cell also for an energy source. So phagocytosis is that idea to eat and bring in that large molecule. Pinocytosis, tiny, I don't know. But it brings in usually liquids or smaller particulates. you'll notice that when it's a smaller thing, it seems to use a vesicle, and when it's a larger thing, it seems to use a vacuole. You can keep that in mind in terms of vacuoles are usually larger and for this kind of storage, and then vesicles are usually more of that smaller suitcase coming off of the Golgi or budding in from the plasma membrane. And then the third kind is receptor mediated. So we haven't completely forsaken the role that proteins can play in the membrane. These are here to basically respond to a signal. We're going to talk about this more in a couple weeks. This is probably going to be class 6. But receptors on the ligand, the molecule that size and shape, and then it invaginates, it forms a vesicle around it, and it brings it in to have basically that ligand end up on the inside of the cell. Okay. So, oh, one other thing I did want to mention there. That was all endocytosis coming into the cell. What do you think would be something leaving the cell? See if anybody already had some questions before. In the interest of time, I'm going to jump ahead. Exocytosis, exiting. So it goes out. Okay, let's practice with a question. And I hope you've enjoyed today's class on the cell. I think it's this beautiful combination of all kinds of specialized things that come together. And then they themselves further specialize. It's really amazing. It goes beyond the idea of just chemicals. The chemicals are going to chemistry. They're going to do their thing and interact and react. But to have bigger things come together and have them play a role like this and cooperate, but still just kind of come out of randomness, that's amazing. Okay, so let's share now. I've talked to you before about our questions 1 and 4 tend to be the ones that are going to have the style the College Board is expecting for the AP exam this year. I wanted you to at least see how the content that we covered would be assessed. So this is question 6 from 2013. The following data were collected by observing subcellular structures of 3 different types of eukaryotic cells. And so you would want to know, what is smooth ER? What is rough ER? What are mitochondria? What are cilia? What are Golgi bodies? And then how do cells differ in what they have of those things? Based on analysis of the data, essentially the table that you see here, identify a likely primary function of each cell type and explain how the data support that identification. So this is still one of those shorter style questions like a number 4. The better you know these things, the more comfortable you're going to be making sure that you demonstrate the knowledge that College Board is looking for. So we have to just simply identify a likely primary function and explain. So I'm willing to bet we're not getting one without the other. So I'm going to give you a minute to think about that, but we are definitely over an hour at this point. Thank you for hanging out with me. I hope you enjoy this as much as I do. Okay, ready? Scoring guidelines. So you get a point for each correct pair. So however many points this question could be, 3 maximum in this case, you'd have to get for 1 point for each pair. Essentially, you can't put 2 or 3 pairs for one cell type. Name 1 for each, and that's enough. So we had a small amount of ER, a smooth and rough, large amount of mitochondria and cilia. You naturally want to be thinking transportation, locomotion, movement, that kind of thing. The energy and a little hair on the outside of the cell, that's the aid stuff, that's definitely locomotion. And when you basically identify that function, you want to explain, okay, cilia for movement, large amounts of mitochondria provide the energy. It's precisely what I said, it's just more formal. Cell Y, large amount of smooth ER, large amount of rough ER, so a lot of synthesis going on, both lipid and protein. A little bit of mitochondria, no cilia, and a lot of Golgi. The big thing here would be that idea of synthesis and then vesicle formation. You're making a lot of stuff, you're shipping a lot of stuff out. This is a factory. That's what this cell is. So secretion and exocytosis, because we're making inside, so we got to send it outside. That's exocytosis, and that's just because of the if we were doing proteins, we talked about the rough ER in that cell, it would be a smooth ER. And then cell Z, it has nothing. What could this possibly be when there are very few organelles, or rather there are none? Any of the functions that are independent of organelles. So the usual stuff of transport or protection or supporting or storing, that's probably what I would have gone with. Golgi bodies are the vesicle formation for export and import, but vacuoles, they don't mention those here. We could still go with storage. And lots of space, so because we don't have any organelles in there. I think this is kind of a very clever style question, but you could also come back at them and be like, there is no function. It doesn't have anything. It could be dead, and you'd be right, and they give you that point. Okay, so if you want, let's go back here. If you want another question for practice, what I would recommend is checking out number 8 on the 2019 exam. You can get these exams at AP Central and College Board. Check it out. Practice. It's always good to study and then assess what it is that you reviewed to make sure that's clicking, and then go on to your practice questions and put it to work. So it's kind of a double set of practice questions there. One with assistance and more informal, and then another where you're really kind of trying it in the style of what you expect in the test itself. That is all that we have today for the cell. Thank you for sticking around. We ended there in a very poetic way about active transport and energy being required. If you were thinking, where does that energy come from? How do I make that? How do the cells make that? Well, then join us next week. PMTV, AP Biology Review, Class 5, the cell and energy. If you have any questions, classes@prepmatters.com, hashtag PMTV. I'm at Mental Markets on Twitter. Send me your questions, and we'll figure this out together. Thank you, everyone, and class is dismissed.